Yes, so thank you, Tom. Thank you, Kathy. Thank you, Paul Allen, for hosting this wonderful event. I want to briefly acknowledge my collaborator, Dan Geschwind, who unfortunately couldn't be here today. So my crazy dream is to stop aging. I mean, in certain ways, I feel it isn't a crazy dream at all. In certain ways, it's a dream that should be driven by market forces, echoing the earlier speaker today. In principle, why market forces? Because it's straightforward. We have all these technologies, we have compounds, and what we could do is we take a bunch of cells, we try our favorite compound, and then we say, look, the cell is rejuvenated by 10 years. It's in certain ways an industrial endeavor. So why aren't people doing it? Well, there is an obstacle, which is we cannot measure aging. If we had an accurate measure of biologic age, we could build a whole industry around finding rejuvenating therapies. So in any event, in my lab, we are interested in a way of measuring biological age, which is called the epigenetic clock. And these pictures demonstrate some of the key properties of the epigenetic clock. So first of all, the epigenetic clock applies to all sources of DNA, all cells, all organs. And therefore, you can measure, for example, the age of a neuron. You can measure the age of the cerebellum. You can measure the age of liver, heart, aorta, bone, saliva. If a cup of urine contains enough DNA so that I can tell you how old you are. So the other property is visualized in the other plot, which is the epigenetic clock applies to the whole lifespan. It applies to prenatal samples, prenatal brain samples, all the way to supercentenarians. So I talk about an aging clock, and I want to give you a precise definition. So for me, the word clock implies a very accurate measure. And so for me, accurate means that if you have the perfect data set, 1,000 people from age 0 to age 100, and you estimate their ages, and this age estimate has a correlation with the true chronological age of 0.8 or more, then I think it deserves the label a clock. Now, it's critical that this assessment is made in completely independent data generated by different labs, because it's very easy to cheat with statistics. So I'm not aware of any biomarker that meets this very stringent criterion, except for biomarkers that are based on DNA methylation levels. I mean, all of us prefer transcriptomic data or proteomic data because it's closer to biology. But nobody has been able to develop an aging clock based on these genomic data. You really need methylation data. So exactly 3 years ago, almost to a day, I published what I call this multi-tissue biomarker of aging. And it was so accurate that the strongest criticism was that it couldn't possibly measure biological age. But I want to show you today that we have sufficient data that really illustrate it does capture an aspect of the root cause of aging. Before I go there, I want to remind everyone, methylation is, of course, a chemical modification of a cytosine in humans. And it's sometimes known as the fifth base. So the epigenetic clock method is implemented in 3 steps. In step number 1, we measure the methylation levels of 353 CpGs that are, can be found on all chromosomes. In step number 2, we form the weighted average of the methylation levels, but that's not yet on the units of years. So in step 3, we apply a nonlinear monotonic transformation and then turn this into an estimate of age, which is referred to as epigenetic age or methylation age. And the really striking part of this algorithm is that it's the same definition for every tissue and cell type. That is really completely surprising to people who study methylation profiles. So notice in step 1, I didn't say you need to specify you're dealing with blood or with neurons. Whatever DNA you give it, it will work. Remember in step 2, I showed you there's a weighted average before you transform it and calibrate it to age. And I want to show you how this weighted average depends on age. So you see that early on, as the organism grows from age 0 to 20, there's this sharp increase. But after age 20, there's an almost linear relationship. And so we say the ticking rate of the clock after age 20 is constant, the rate of change is constant, but before it, during growth, the ticking rate is fast. I want to also highlight for this audience that it's exceedingly accurate in brain tissue, in every brain region, and also in sorted cells, for example, in glia or in neurons. And the latter is important because neurons are post-mitotic. And therefore, we know that the epigenetic clock that is so accurate in this post-mitotic cell type does not measure mitotic age. It's an independent process. In the lower bar plots, I show you the ages of different brain regions in middle-aged people, and notice the temporal cortex has the same age as frontal cortex and the cerebellum. In middle-aged people, all brain regions have the same age. However, we recently analyzed the brains of centenarians and supercentenarians and many other body types, and the striking thing was that the cerebellum was by far the youngest part of the body. So if you ask me what part of the body ages slowly, it is the cerebellum. That has been replicated over and over. So I'm here. So we have this very accurate tool, but the major gap is that we don't understand the biology really that underlies that tool. And here I want to show you that most recent data that show that it plays a role actually not just in aging but also in development and neuronal maturation. So to begin with, I show you data from Jonathan Mill, who showed that the epigenetic age of fetal or prenatal brain samples correlates with the actual age. You notice the actual age is negative, which means prenatal, and the units are years. That is right. Normally one would want to plot gestational week. The fortunate thing about these Allen Frontiers meetings is one gets to know one's colleagues. So Bill Lowry from UCLA attended the last Frontiers meeting, and we got talking. And we said, why don't we apply the epigenetic clock to his cell model? And strikingly, we found a very interesting effect. So iPS cells always have negative ages, perfectly young. Then his neural precursors were already older. And then the more differentiated neurons were even older. Notice the age estimates are all around 0. So it recognizes these are still young cells. But for me, it's stunning that cell differentiation status really tracks epigenetic age. The other person I met at the last Frontiers meeting is Thomas Ray. And we also felt we should apply the epigenetic clock to his fetal retina samples. And for me, this was the most striking result in the last half a year. I mean, the result is completely shocking to the extent that I feel the data are made up. So but let me walk you through it. These are fetal retina samples, and the x-axis is really gestational week. In units of weeks, it would be week 16 to maybe 40 or 30. And on the y-axis, we see the methylation age estimate of the fetal retina. And here's the stunning thing. That's a correlation of 0.89, I mean, so clearly this, the epigenetic clock must have a purpose. There must be there must be an innate process that plays a crucial role in development. And then later, as we age, it's unarrested development, and it plays then a role in aging, as I will show to you later. So I briefly want to mention that when you start from a cell and you turn it into an iPS cell, you completely reset the age. You may start with the primary cell of a 50-year-old, and then the iPS cell will have a negative age. So the question then is, how do we develop a cell model of older neurons? You know, we can't just start with fibroblasts and turn them into iPS and then differentiate them. That's, of course, the purpose of our grants. So I want to show you a recent publication from Andrew Yu and Christine Hu, where we showed that microRNA-based neuronal direct conversion of fibroblasts into neurons maintains the epigenetic age. So just to walk you through it, when we start with fibroblasts from different people, and we administer these microRNAs, we turn them into medium spiny neurons, and this graph shows that the methylation age of the fibroblast is completely preserved in the resulting medium spiny neuron. Because Halloween is coming up, you may be wondering, what does the Grim Reaper really look like, you know? So a journal offered me the opportunity to create a cover, and I sent them that, and they said, this is not serious enough. So they took this part, they took the molecule, everything else gone, you know, talking about artistic freedom. I was very offended, but in my opinion, the Grim Reaper looks like a methylation modification, and I tell you why. So we have conducted a large-scale meta-analysis. It just was published 3 weeks ago. It has a huge number of co-authors because it involved 13 different cohorts. We had 13,000 people. Of them, 2,700 had died. And so we had methylation data from the blood data from the 90s, and we could see, can we predict their lifespan? And the answer is yes. And these p-values are extremely significant. So we have several different measures of epigenetic age acceleration, and the p-values are 10 to the minus 11, 10 to the minus 9, and 10 to the minus 43. No matter how you measure epigenetic age acceleration, it does predict lifespan. What is even more stunning is it predicts lifespan after you adjust for all of these potential confounders: chronological age, body mass index, educational level, alcohol, smoking, history of diabetes, cancer, hypertension, physical activity. Everything you can think of that could confound the relationship, you adjust for it, it remains a significant predictor of lifespan. Again, pointing us to the idea that the epigenetic clock relates to an innate, genetically determined aging process that, as I showed you earlier, matters also for development. People ask me, what has the strongest effect on epigenetic age? The strongest effect is trisomy 21. So when we look at blood or brain samples from Down syndrome subjects, that's where we see the strongest age acceleration. I want to show you some unpublished data where we applied the epigenetic clock to dermal fibroblasts from Hutchinson-Gilford progeria. This is data from the Progeria Research Foundation. So to do that, I developed a new epigenetic clock that's really tailor-made for fibroblasts, because many of us work with fibroblasts. And it does not correlate with cell passage number or doubling level. So in any event, here's the punchline. So I applied the epigenetic clock to children, because progeria cases unfortunately die very young. And the points are colored by disease status. And you see there's actually no difference to control samples in these children, you know. So according to the epigenetic clock, the dermal fibroblasts of these progeria subjects do not differ from controls, with the exception perhaps of what is known as non-classical progeria. There we see a little bit of a hint, you know. So to me, that means that the epigenetic clock is upstream of these, the progerin molecule, you know. So when you think of it as a causal chain. Yeah. I also want to briefly mention, we analyzed the epigenetic age of the prefrontal cortex, and we found that this epigenetic age relates to neuritic plaques, amyloid loads, Alzheimer's disease, and even pre-mortem cognitive assessments, and we used the religious order data for that. So as part of the Allen grant, Allen Frontiers grant, we conduct GWAS studies to find SNPs, or genes that drive epigenetic aging. And so these data are from Akalu, where we analyzed the epigenetic aging in the cerebellum as a quantitative trait, and then we used SNP markers to see which drive these age acceleration. And we found very interesting genes, a gene that's part of the mTOR complex. We also applied this technique to study aging in the prefrontal cortex. We found a very significant hit. We also pool multiple brain regions. And in total, we have now 4 genome-wide significant hits, and we carry out this expression QTL study to find the underlying genes that mediate the effect. And then as part of this Allen Frontiers challenge, we follow these genes up by CRISPRing them out and validating them in the dish. By now, many phenotypes have been linked to the epigenetic clock, from Alzheimer's disease to obesity, arthritis, Parkinson's disease, Huntington's disease, and so on. So in summary, I feel the epigenetic clock captures aspects of biologic age. It excitingly tracks also neuronal maturation. It's not the same, but it correlates. And I should highlight user-friendly software can be found on my webpage. I want to thank again the Paul Allen Frontiers Group, Dan Geschwind, and many others. Thank you so much.